For decades, researchers assumed that a larger brain automatically meant more brain cells, and by extension, greater intelligence. But measuring the actual number of cells in a brain was exceptionally difficult until neuroscientist Susanna Herculano-Houzel developed a new technique called the isotropic fractionator. By dissolving brain tissue into a uniform liquid suspension, a process colorfully described as making brain soup, researchers could precisely count individual cell nuclei under a microscope. Herculano-Houzel's work revealed that the average human brain contains roughly 86 billion neurons. More importantly, it showed that human brains scale along the same rules as other primates. We do not have a specialized exception to cellular biology. We simply have a large primate brain built with consistent physical scaling rules. This cellular insight suggests that raw physical volume is a poor way to measure mental capability. Instead, cognitive power depends on neural density, white matter connectivity, and wiring architecture. Over the last few thousand years, our brains may have reorganized, tightening the physical networks that link different regions. A smaller, more compact brain can process information faster because electrical signals have shorter distances to travel. It also runs on a much lower metabolic budget. Rather than representing a loss of intellectual capacity, a modest reduction in size might represent an evolutionary upgrade, a transition to a highly optimized neural network that delivers the same computational power at a fraction of the energetic cost. Across all mammals, raw brain mass explains only 10 to 20% of the variation in cognitive performance. To compare brain power across different species, scientists often use the encephalization quotient, which measures a species' actual brain size against the size expected for its body weight. While humans have an exceptionally high quotient of about 7.5, other animals challenge any simple link between volume and intellect. Bottlenose dolphins, for instance, possess a quotient of 5.3 in complex societies, yet their cognitive profile is vastly different from ours. Sperm whales possess the largest physical brains on Earth, weighing up to 9 kilograms, but this massive volume does not translate into a proportional leap in abstract reasoning. The disconnect becomes even clearer when looking at birds. New Caledonian crows and African gray parrots have brains that weigh just a few grams, representing a tiny fraction of typical mammalian brain mass. Yet these birds regularly outperform many primates in cognitive tests. Crows can manufacture multi-step tools from leaves and solve complex sequential puzzles without any training. African gray parrots can perform exclusion-based logical reasoning and grasp abstract concepts at a level comparable to a 5-year-old human child. Their cognitive ability is driven by neural density rather than physical volume. Songbirds and parrots pack up to twice as many neurons into the same volume of brain tissue as primates do, achieving high-level computational power without the burden of a heavy skull. Even among modern humans, the assumption that a larger skull equals a sharper mind fails under close examination. Large-scale neuroimaging studies, including analyses of over 1,000 individuals from the Human Connectomy Project, reveal that the correlation between brain volume and IQ is remarkably weak, hovering between 0.18 and 0.20. Once researchers apply controls for height, biological sex, and socioeconomic factors, this small correlation shrinks further. Instead of bulk volume, cognitive differences depend on microstructural features. The degree of cortical folding, the depth of the brain's grooves, the thickness of specific regions like the prefrontal cortex, and the efficiency of white matter pathways connecting different areas dictate how quickly and effectively the brain processes information. Evolution favors connection speed, neural density, and network efficiency over raw physical size. Modern lifestyles introduce an entirely new set of variables. From processed diets to constant screen exposure, our environment has changed rapidly, raising questions about whether these daily pressures are actively reshaping our brains today. To study these potential shifts, researchers look to massive modern datasets rather than ancient fossils. The UK Biobank, which holds health and brain imaging data for tens of thousands of individuals, provides a detailed look at living populations. Within this vast cohort, researchers have identified certain correlations, such as an association between a higher body mass index, or BMI, and lower total brain volume. It is a striking finding, but it is one that requires careful context. When scientists analyze these massive datasets, they find that biological sex and age dominate almost all variations in brain volume. Other potential drivers, such as childhood nutrition, historical exposure to infections, chronic psychological stress, and constant digital multitasking, remain highly debated hypotheses rather than established facts. Isolating the physical impact of any single factor in a modern population is exceptionally difficult. Even within the UK Biobank imaging team, researchers debate how to interpret these signals. Early life infection history and childhood nutrition both leave lasting imprints on adult brain volume. Yet these historical variables are nearly impossible to disentangle from modern lifestyle choices. The biggest scientific hurdle is the difference between cross-sectional and longitudinal evidence. Much of our modern imaging data is cross-sectional, meaning it captures a snapshot of different individuals at a single point in time. While this can show a statistical association, it cannot track how an individual brain changes over decades, let alone across generations. To prove that modern lifestyles are causing a real population-wide anatomical decline, scientists need robust longitudinal studies that track the same families over many decades. Without this long-term data, a correlation between a modern lifestyle factor and a slightly smaller brain volume remains just an observation. Present-day environmental factors clearly shape our individual brain health, but current evidence does not support the idea of a rapid generational physical decline. The current scientific consensus on changing human brain volume is marked by extreme caution. Most neuroscientists and evolutionary biologists view the reported anatomical decline over the last several thousand years as a modest physical shift with highly restricted interpretive value. It is not an evolutionary step backward. Instead, contemporary research has moved away from bulk cranial measurements, focusing instead on the layout of neural networks, synaptic connectivity, and metabolic economy. While theories like self-domestication, dietary trade-offs, and distributed cognition offer plausible ideas, they remain underdetermined and unproven. No single hypothesis explains the entire physical record. Moving the debate forward requires a new class of evidence. Scientists need larger, more securely dated fossil samples from a wider range of geographic regions, paired with standardized body size reconstructions to ensure accurate scaling. Settling these questions also demands long-term longitudinal MRI studies that track human cohorts across decades, ancient DNA links tied directly to neurodevelopmental genes, and precise metabolic models that balance calorie intake with active neural costs. Until this data is gathered, the physical evidence remains an open, fascinating scientific puzzle. This ongoing uncertainty also carries a serious ethical responsibility. The history of physical anthropology is stained by scientific racism, where biased skull-size claims were weaponized to establish false, politically motivated hierarchies of human intelligence. Modern genetics and neuroscience have thoroughly dismantled those claims, proving that bulk brain size is a flatly useless tool for ranking human minds. Brain volume is not a measure of human worth or cognitive potential. The subtle physical shifts in our evolutionary past are not a story of mental decline, but a reflection of how dynamically our species adapts to a changing world. Ultimately, our biological story isn't one of decline, but of constant, brilliant adaptation. As we build a world that requires less raw processing power and more specialized connection, our brains will continue to reshape themselves. The true measure of human intelligence has never been vault space. It is how we adapt. What do you think our minds will look like next? Let us know in the comments.